Well, thank you. I was really thrilled when Ruth invited me to come speak. We were trying to figure out when I was last here, and maybe it was 2017, I don't know. It's been a while, so it's really nice to come back and see familiar faces and meet new people as well. So today I'm going to try to go through some of the last 10 or so years of work that I've been doing in seagrass beds. I've always had a great love of seagrasses, did my dissertation research on seagrasses, and kind of picked back up on doing seagrass work back around 2010. And so I don't know that I have any really big take-home messages, but hopefully we'll find out some interesting things along the way. So a big part of what I'm going to be talking about is some of the research that I've done, but particularly work that has been done by 3 of my graduate students who've done their thesis research in seagrass beds, Rachel Presley, Mackenzie Rothfuss, and Lacey Bowman. So you'll see their names coming up later in the presentation. Most of you are probably quite aware of why we care about seagrasses beds. If do we have any fishermen in the audience? Anybody like to go catch fish? a lot of the commercially important species along the Gulf Coast spend some amount of time in seagrass beds, and so it's important habitats for foraging, nursery grounds. And really, in many of our shallow coastal embayments, they're a major source of primary production that then it forms the base of the food web. And in recent years, there's been really increasing interest looking at carbon sequestration and their role in terms of blue carbon. So I'm going to talk primarily about the nitrogen cycle and a little bit about sulfur cycling today. So we're not really going to cover the habitat side of it. Probably most of you are also pretty aware that there have been global losses in seagrasses, a variety of factors that contribute to this. Some of those major factors generally include habitat destruction, and it may be both direct losses due to dredging that occurs, but often it's more indirect, sometimes due to degradation of water quality and other things that affect how well the seagrasses can grow. Variety of other factors that also contribute to losses of seagrasses. And one of the little things I wanted to mention here that I'm going to come back to in the talk is the role of sulfide. Hydrogen sulfide is a phytotoxin. It can destroy the seagrasses in high enough concentrations. And there's been a variety of work done on sulfide in Florida Bay. So we're going to talk a little bit about some work that we did in Pensacola Bay related to sulfide. So 3 dominant seagrass species. Of course, there's a lot more freshwater species that are found up in the creeks and the river deltas. I'm going to focus on the marine side of this. So our dominant species, Thalassia testudinum, climax species, long-lived, very thick meadows. Haliduli reichii is the next most. Well, both of those are dominate under sort of different conditions in Pensacola Bay. Cuban shoalgrass, and then Rupia maritima, which we find in a wide variety of habitats throughout Pensacola Bay. It can be very dense beds in more of the mesohaline parts of the estuary where it has some protection to form beds. But we will find it intermixed sometimes with Thalassia and Haliduli. So I'm going to start the talk by giving a little bit of a historical perspective, talk a little bit about the Pensacola Bay system, very briefly talk about some of the factors that influence seagrasses, and for the rest of the talk, kind of focus on the role of nutrients and how that may affect productivity, some productivity work we've done, and some nutrient cycling work that we've done, and then kind of ended a little bit on this sulfide story and what we know about sulfide in Pensacola Bay. So I would maintain that Pensacola Bay is somewhat characteristic of a typical Gulf of Mexico estuary. So if you've not been to Pensacola Bay, you need to come and go to the beach. We have beautiful white sand beaches, crystal clear waters, great place. So this is my plug for the tourism part of Pensacola. The watersheds of the Pensacola Bay system extend up into Alabama. And as you can see from the graph on the right there, we have major freshwater coming in along the western side of the watershed through the Escambia River. And then on the eastern side, we have Blackwater and Shoal rivers coming in. And that map on the right-hand side there is color-coded. It's kind of a dated map. It's a 2001 land cover, but there is increasing development throughout the watershed, particularly in the coastal zone. So you can see from the colors there that those reds and yellows are the higher density developments. We do have some amount of agriculture in part of the upper watershed, a lot of forested lands, and a lot of silviculture in the upper parts of the watershed. And this bottom photograph is a photo from Fort Pickens right there at Pensacola Pass, which shows you that it's kind of got an industrial character to it as well. So like many estuaries, again, not unique to Pensacola Bay, we have seen increased urbanization and development within the region. And a lot of the increase in population, increase in development, increase in urbanization, commercialization occurred following World War II. We saw a rapid increase in population. We saw a lot of industrial activity, a lot of chemical plants. And as a result, we have a lot of the issues that many estuaries face in terms of contaminants and industrial discharges. We had a coal-fired power plant. Now it's gone to natural gas, so that's a good thing. We had a major PCB spill in 1969, which still has some remnant PCBs that we can find in the bay. We have a paper mill. We have a lot of Superfund sites as a result of contamination, and this industrial contamination, and then coastal development. As I said, beautiful white sand beaches, clear waters. This is a prime area where people want to come and live. And because we have a lot of military bases in the area, a lot of people have sort of come through this region for training, and then when they retire, decide this is the perfect place to retire, or decide that they don't want to live in Michigan or Wisconsin when it's cold all the time and come live in Florida. So generally quite an affordable area for people to come live in, and so coastal development is very, very high. Not surprisingly, we also have other kinds of major things that have happened. The Deepwater Horizon oil spill, did have some effects in our system, but really quite minor compared to the major damage suffered by Louisiana, Mississippi, and of course you guys. We did get some oil on our beaches. We probably got some oil in the bay, but not a huge amount of not the level of negative impacts that we're seeing west of us. One big consequence of that, though, is all of the money that was put towards monitoring, research, and education as part of the Restore Act funding. So some of the things that I'll be talking about today have sort of come about because of the interest in understanding what happens with water quality, realizing that as horrible as Deepwater Horizon was, we're actually doing a lot based on our day-to-day activities that can have negative impacts on our estuarine and coastal environments. So, so the Restore Act is given us a mechanism to try to address some of these more long-standing anthropogenic impacts into estuarine systems, estuarine and coastal systems. So what about seagrasses? If only we had a time machine, it would be great. It would be so cool to go back in time and see, what the bay looked like historically. I have a postdoc who's really interested in historical ecology. And of all the things that, early people wrote about, usually didn't do a lot of writing about seagrasses. So you get little clues here and there, grassy point, probably an indication there were seagrasses there, but really not much else. So we're really dependent on not really knowing much about seagrasses until about the 1960s. And this is a period when people were flying airplanes and were taking photographs out of airplanes. And so you can make some assumptions about where seagrasses were. And so these references that are on the graph are taking some of this historical data from the 1960s and to some extent, mainly the 1960s data, 1950s and 60s data. There was no ground truthing done of this data. It's based on kind of anecdotal reports. And so this is what we think was going on during that period in the Pensacola Bay system, with Santa Rosa Sound, that white bar, being the location with the greatest seagrass beds, and then other important locations throughout the bay that really disappeared between 1960 and 1980. So many of those areas in the mesohaline parts of Pensacola Bay, really lots of issues that led to losses of those seagrasses. But we still do have seagrasses in the more marine parts, in the Santa Rosa Sound and Big Lagoon areas. And there are some indications that there may be other places where seagrasses are starting to come back. So there is a little bit of a good news story here in terms of we're not seeing continued losses of seagrasses in our system. And this is a map from one of the first environmental reports, of a comprehensive report done for the Pensacola Bay system, where basically what they're doing is they have drawings of that shoreline along the Gulf Breeze Peninsula, and the bottom one is the oldest one, and then you're going forward in time. And so they think back in maybe the 1960s. There was thalassia and haliduli beds that were all along the lower part of the Pensacola Bay system, and that persisted maybe for a year or two, and then by the time you get to 1975, there was none of it left in the system. So this is the kind of quasi-quantitative information that we have about seagrasses, early information about seagrasses in the Pensacola Bay system. This is another report where, again, they're doing interpretations and trying to understand what's going on with seagrasses. In recent years, we've been able to do more quantitative and more thorough surveying, and the folks down at FWC have done a great job with the help of a lot of collaborators, including Dottie and Ken Heck, who've done a lot of the surveying in our Pensacola Bay system, looking to see where seagrasses are. And so they have now put out 3 reports that have done these surveys. So those are really the best recent information. So this was the first one was put out in 2010. So really starting to get a better handle on what's going on. And we have collaborated with the FWC group doing work in 2016. They had a project called Roadblocks to Seagrass Recovery. And this was a Restore Act funded project. And we did several things, and I'll show you a couple of them later in the talk, but this was one of them where we did some surveying to kind of see where there were seagrasses in different parts of the Pensacola Bay system. So all the different colors are telling you which species you find, and there's mixtures of species in different locations. So for today, I'm going to really focus my attention on the two locations in the Pensacola Bay system where we have our greatest abundance of seagrasses. To the east is Santa Rosa Sound. The north side of it is the Gulf Breeze Peninsula, and the south side of it is Santa Rosa Island. And then just to the west is Big Lagoon, and Big Lagoon is connected to Perdido Bay through the Intercoastal Waterway. So it's very close to the pass. It may have some influence from Lower Pensacola Bay and also from Perdido Bay. So those are two main locations where most of my seagrass research has been done. So what is it that makes seagrasses healthy? What do they need? Well, like most estuarine organisms, they kind of have a preferred salinity zone, and so in our area, we have that sweet spot for salinity in Santa Rosa Sound and Big Lagoon. That's that kind of mid-range of salinity, anywhere between maybe 18 and 25, 30. That's really great for seagrasses. They don't really like a lot of fresh water. They don't like hypersaline conditions, but we're not Laguna Madre, so hypersalinity is usually not a big issue here. One of the major factors that really controls seagrasses is light. They're living on the bottom. They're growing up from the sediments. They can't move. They're stuck there. So whatever the light environment is, that's going to have a really big impact on how well they grow, because they need light for photosynthesis. So the things that can affect that light level are turbidity, particulates in the water. Also, we have a lot of color. If you're familiar with the term blackwater rivers that have tannins and other organic constituents, that color in the water can also have a negative impact on light availability. And then biological factors also can limit light. If there's a lot of chlorophyll in the water, you have a lot of phytoplankton, single-celled algae growing in the water, they're going to be the winners. They're capturing the light, and the seagrasses aren't. They're going to get to it first. And then finally, you can have epiphytes that are growing on the leaves of the seagrasses. These single-celled microalgae and sometimes macroalgae growing on them. Again, if they're growing on the surface, they're going to be the winners and the seagrasses are going to be the losers. So light is absolutely critical for seagrass habitats. And I'm not really going to talk much more about light availability. There's some interesting things that we're trying to do in recent years related to that, but I'm not going to talk about that today. I'm mainly going to focus on nutrients. And so again, seagrasses have an option for where they get their nutrients from. Their leaves are up in the water, so they can have leaf uptake of the nitrogen and phosphorus into the leaves, or they can get nutrients from pore water. The roots go down into the sediments, and so you can have pore water uptake. So we'll talk quite a bit about that. As I mentioned, we're going to talk about sulfide and sulfide production in sediments and how that can be toxic. And then I couldn't come here and not talk about bottom-up controls. I got this backwards. The importance of grazing on seagrasses. So not work that I've done, but grazing effects on epiphyte communities and trophic level interactions and disruptions to the different trophic levels can also play a really huge role on how well seagrasses do. So this is my little cartoon of the seagrass bed. So for this talk, I'm going to talk a bit about carbon fixation. So how we have production of seagrasses. I'm going to talk a little bit about nutrient uptake, both from the water column and the sediments. And then some studies that my students have done on nitrogen fixation work. So how you get new nitrogen into the system. And then a little bit about the decomposition side of it. And I'm not really going to spend any time talking about the other parts of the nitrogen cycle. There's a lot more that could be done on this, but I didn't want it to go too long. So what kind of measurements do we have of primary production in the Pensacola Bay system? So there's a variety of different techniques that you can use. The box plot over here is a summary of measurements that have been made in the Pensacola Bay system. Two of the sets of measurements, the two on the left, the Hester et al. And the 2022 domes, were done by putting chambers down over top of the seagrass beds, and that's what's pictured in the photograph as a chamber with a stirring system on it. And so that gives you, some estimates of what gross primary production is when you have chambers that are in the light and also in the dark. You can use that to estimate primary production. The Merle et al. Paper from 2018 estimated benthic production in a slightly different way. This was looking at oxygen measurements, oxygen in the water column. And also he made a set of measurements of water column production and respiration. And so the sensors that were out kind of gives you the total system production. And then he subtracted out the water column production to get an estimated benthic production. So you see there's some pretty big error bars on that, but still, the median is pretty much in line. So, I think that's pretty, it's not an unreasonable approach. And then finally, we are working on a paper that is hopefully coming out soon, doing a kind of a classic technique where you look at the accumulation of biomass. You actually poke little holes in the seagrass leaves and come back, 10 days later, 12 days later, and harvest all the seagrasses and see how much grew. So you can see where the poke is, and then you can measure the biomass accumulation. So this is above ground biomass, and again, it's pretty much in the same range. Again, all the dome work was done with oxygen, so you have to convert oxygen to carbon. How you do that? If you're interested, I can talk to you about that. But just general point is, I think we have a reasonable indication of what primary production is in our seagrass beds, and it seems to be fairly consistent across these different methods. This is a little bit more detail about this leaf marking technique. And so we had a variety of different sites. So this work was part of the FWC Roadblocks to Seagrass Recovery project. We looked at it at a variety of different locations in Santa Rosa Sound, in June, July, and September, and we don't have a huge number of measurements in July, but it looks like that's really more the peak period, and September's a little bit lower. There are some station differences, but not any real straightforward or consistent patterns in that. Another kind of indirect way to look at how well the seagrasses are doing is looking at accumulation of biomass in the seagrass beds. And in 2017, I started a citizen science seagrass monitoring program. And this gets volunteers out to survey seagrasses, and they pick a location and they try to go back to their same sites and look at how much cover there is. And again, it's citizens, there's varying amounts of data in this. This is all we have multiple sites in Big Lagoon State Park. So it's including a variety of different sites, but it does kind of, if you squint a little bit and look at it, it does sort of suggest that you get your kind of maximum biomass in July, maybe August. So that's you getting your production, seagrasses are increasing, you get more cover. And the peak is kind of in that mid to late summer, and then September and October, maybe they're hanging on, maybe they're dying back. So that's what we kind of know about productivity in seagrasses. What about where they're getting their nutrients from in order to take up those nutrients for growth? So we've made a wide variety of measurements of nitrogen and phosphorus. These are dissolved inorganic nitrogen, so both nitrate and ammonium, and dissolved inorganic phosphorus at the two major locations, Big Lagoon and Santa Rosa Sound, both what's in the water and then lots of measurements down in the sediments. And I'm going to go through the sediment measurements in a little more depth, but just wanted to give you some ranges. So Pensacola Bay, like many other locations, much higher concentrations in the pore water. It's not a surprise, not anything unusual. This is where decomposition is going on, organic matter that's down in the sediments, getting broken down, releasing that ammonium phosphate. You may get some nitrate if you have nitrification going on. In the water column, there's lots of competition for those nutrients. You have phytoplankton, you have seagrasses that are taking it up. And in general, because you have more water from the Gulf of Mexico, the water that's in the environment naturally has relatively low nutrient concentrations. So if we look at our nitrogen levels in the two locations, we see the averages are pretty similar. We occasionally have some high nutrients in Santa Rosa Sound, but generally pretty low, and often a little bit more ammonium than nitrate. Phosphate levels are similar and quite low in both systems. Pore waters are higher, and again, that's to be expected. These are very sandy sediments, and these values are a little bit low, but not out of the range of what you often find for pore waters and sandy sediments. If we were talking about muddy sediments, these DIN and DIP concentrations might be 10 times higher. So, so again, pretty similar concentrations of pore waters on average. Again, we do have some high levels in Santa Rosa Sound. And then I'm going to specifically talk about Rachel's thesis research and what my student Lacey is working on for her thesis in terms of nitrogen fixation. So those are the major nutrient sources. So if we look at this sort of accumulation of pore water ammonium data by month, and I haven't really separated it out by the two stations, based on whether it's from bare sediments, so there's no seagrasses, the haliduli sediments in the triangles, and the thalassia sediments in the diamonds, we do see that that bare sediments actually often have higher nutrient levels than the seagrass sediments do. And this is particularly noticeable in the summer months. And this really makes sense because what do we say? We have our high growth period. It's in the summertime, right? So they're going to have a big nitrogen demand. And so they're able to suck down the nitrogen that's in the pore waters. And that leads to lower concentrations. In contrast, in the circled area, we see some quite high nutrient levels starting in September and October. And this is likely a result of this decomposition that's going on. So in the grass beds, these grass beds are accumulating dead leaves, they're accumulating other sorts of organic matter. All that organic matter is kind of settling out in these grass beds. All the microbes are very happy. We still have warm water temperatures in September and October, so a lot of decomposition going on, a lot of remineralization. So we're getting production of ammonium, production of phosphate. And because the seagrasses are kind of slowing down for the season, there's not as big a nutrient demand. And so we get this accumulation of nutrients in the pore waters during this period of time. And then by winter time, January, February, generally pretty low concentrations occurring in the winter months. So, so one of the things that one of my students was really interested in was what role bioturbation plays on sediment biogeochemistry. She really wanted to study the rays, but she was in my lab, so she had to do biogeochemistry. It's kind of a requirement if you're in my lab. You have to do something related to biogeochemistry. So, so she was actually had worked with me as an undergraduate, and so she thought it would be interesting to look at this in the haliduli sediments, which haliduli is a little bit more delicate of a plant compared to thalassia. Thalassia is really pretty stout, pretty thick. kind of hard for the rays to get in there and really do much of anything. The Thalassia, it's much easier for them to kind of get in there and excavate sediments. They're looking for the benthic invertebrates in the sediments. So the photograph on the right shows what happens in a ray pit, that the ray has kind of nestled down in the seagrass bed. It's used its pectoral fins. It's basically vacuuming up, resuspending the sediments, vacuuming up any kind of invertebrates and can get out of the sediments, and it leaves this little depression. So she was following what happens in these ray pits, over time, and doing that by pulling pore water samples to look at what's happening with the nutrient levels. So this is a figure from her thesis, and she was looking at what was happening both in the surface sediments, which is the one with the little 2 on it. So that's the surface layer, 0 to 2 centimeters, and the 10 is the 8 to 10 centimeter layer, so kind of down deeper in the root zone. She had 3 different locations, and the circles are in the ray pits, and then the triangles are in the haliduli bed right adjacent to the ray pit. And so what you can see is that actually, like we saw with some of the other data, higher nutrient levels in those bare pits, and in the seagrass beds, we had lower nutrient concentrations. And we think that this, actually, the effect of having the rays in there, in addition to removing all the benthic invertebrates, they're also creating this pit structure that serves as a little like mini deposition center. And so all of that organic matter can accumulate in the pits, leading to higher nutrient levels, which then can present a nutrient source for the plants to kind of grow back in and recolonize the pits. So moving from, actual nutrients to process, so we have some understanding here of what's happening with pore waters. We have ammonium and phosphate levels. If we look, this is kind of taking that earlier graph on ammonium, the monthly ammonium graph, and then dividing the ammonium concentrations by phosphate. And that horizontal line is at 16. So if your nitrogen to phosphorus ratio, that's 16, that's kind of a standard Redfield ratio. If you look at the relative proportion of ammonium and phosphate in the sediments, again, with the same kind of symbols, the bare sediments being those circles, it's almost always above 16. And all of the seagrasses are well below 16. So what this means is that they're very efficiently taking up any of that nitrogen that's available, and perhaps at some point there's not enough nitrogen there. They may end up becoming nitrogen limited. So how do they deal with that? What can they do? Well, the seagrasses themselves can't do it, but we can have the microbial process of nitrogen fixation. And so nitrogen fixation is a way to take atmospheric nitrogen, dinitrogen, 80% of what we're breathing right now, and turn it into a form that is then usable by other organisms, i.e. primary producers. So the microbial process fixes dinitrogen into organic nitrogen, and that can be passed along to the seagrasses. So Rachel came to me and was very interested in nitrogen fixation. She was actually a Ken Dunton student as an undergraduate, and had done some field work in seagrasses. And so she wanted to look at nitrogen fixation. And so she went out and did a annual study looking at nitrogen fixation at one of our sampling sites in Santa Rosa Sound, comparing sediments without seagrasses, those with Haliduli wrightii, and those with Thalassia testudinum. And so this box plot kind of summarizes all of her data, all the seasonal data. I'm not going to show all the seasonal data, But what you can see is that we see this enhanced nitrogen fixation occurring within the seagrass beds. So the fact is that it's an ability of the nitrogen fixing bacteria to take advantage of organic matter that's in the seagrass beds to do nitrogen fixation, which energetically is a very expensive process. It's not something that, you know, nitrogen fixer is just going to do. It takes takes 16 ATPs to fix a mole of nitrogen. So you're not just going to do that just sort of willy-nilly. So you need to have enough energy to do that. So having that extra organic matter in the seagrass beds is definitely an advantage. So we see higher rates in the seagrass beds, and she also found that there were some interesting relationships between the nutrient levels in the pore water and the nitrogen fixation activity. So this is taking her data and averaging it by season. And what we find is that, again, the bottom line with the ammonium to DIP ratio, that's what's in the pore waters. We see in the bare sediments the same pattern we saw in the previous slide, where the N to P ratio was well above 16 in the bare sediments. And in the grass beds, it's well below. And overall, nitrogen fixation rates were lower in the bare sediments, but in the seagrass sediments, we have this suggestion that we have higher nitrogen fixation occurring when those nitrogen levels are particularly low. So, so this is kind of the concluding slide of her thesis research, which we got published a couple years ago, showing that if we look at in more depth at what's happening to stimulate nitrogen fixation, the correlation matrix that she did suggested that the highest nitrogen fixation occurred when N to P levels were low in the haliduli beds. Surprisingly, in the thalassia beds, wintertime had the highest nitrogen fixation, and that we're still not quite sure why, but that's kind of what came out. And in the bare sediments, it looks like there was some relationship between the nitrogen fixers and the sulfate reducers, where we had high hydrogen sulfide in the sediments and high nitrogen fixation. So my current graduate student, Lacey Bowman, is also interested in nitrogen fixation, but she's looking at it as to what's happening on the leaves. Now, I mentioned earlier that we have these epiphytes that are growing on the surfaces of the leaves. And if you look at the images over there on the right-hand side, those are thalassia blades that she scanned, and you can see they look kind of fuzzy. So she's looking at the chlorophyll content. She's looking at some other pigments. She's also measuring rates of nitrogen fixation. And one of the really cool things that she's doing is she's looking at the abundance of the gene responsible for nitrogen fixation. So NIF-H is a critical component of the nitrogenase gene, and it's what's often used to sort of indicate, whether you have nitrogen fixation or not. So, so she's been doing a lot of work trying to optimize, the primers and trying to get everything working, which those of you who do molecular work know it seems cookbook, but there's an art to it as well, which not my work. I don't understand it, but I recognize the art in it. So, so she's been doing a really good job kind of getting that stuff together. And so she was really interested in comparing locations in our areas where we have a lot of coastal development, which are the two locations on the right-hand side, with two areas that are kind of conservation areas, near the Gulf Islands National Seashore. So not a lot of development immediately adjacent to it. So she's been measuring rates of nitrogen fixation in the two different species. And this is her preliminary data from last June. She just did her last set of experiments yesterday, so she is most excited. So this is a work in progress, and I'm really looking forward to kind of figuring out how this whole story is going to work out for her. So that's the nitrogen fixation story. So just kind of summarize, pore water nutrients, very important. Nitrogen fixation appears to be important both in the sediments as well as in the on the leaves. So now a little detour into sulfur cycling, most of you, if you have done any kind of field work, have probably encountered hydrogen sulfide. It's that rotten egg smell that you smell out in the marshes. So it's a result of this dissimilatory sulfate reduction. You have accumulation of sulfide, hydrogen sulfide. It can very rapidly be oxidized. So sulfide generally doesn't stick around very long, which makes it a little bit of a problem to measure. It's hard to really do justice when you're trying to measure sulfide concentrations. So as I mentioned earlier, the Florida Bay work really suggested that sulfide has a real negative impact on seagrass survival when it's coupled with both high salinity and high temperatures. There's other work as well that has shown its kind of phytotoxin effects. It's very rapidly oxidized, so it doesn't stick around if there is oxygen present. So once you get to the surface of the sediments, there's usually oxygen in the overlying water that will rapidly oxidize it. If you have plants that put their roots out and those roots are releasing oxygen, then they're creating a protective zone around their roots. And if you've worked in wetlands or in seagrass beds, sometimes you take a core and you can actually see that kind of rusty color surrounding the roots. That's because they're putting oxygen out around the roots. So in seagrass beds, the thinking is they can release oxygen from their roots, and because the grasses are super saturated in the water, that also provides some amount of protective effect for the grasses. We have not done quite as many sulfide measurements as ammonium and phosphate, but we have done quite a few. And so this is our summary of pore water sulfide concentrations, again by month. And again, you'll notice really high concentrations in the summertime. And when Rachel started measuring pore water sulfides for her thesis research, we were a little bit taken aback because the folks down in Florida Bay kind of said 3,000 micromolar was kind of where you start killing off seagrasses, and we certainly see those numbers, and we don't see like big die-offs of seagrasses. So what's going on? So one of the other parts of our FWC project was trying to specifically do an experiment to look at how sulfide might affect growth and production of seagrasses. And so the idea was that we were going to do an experiment in the thalassia beds and the haliduli beds in Santa Rosa Sound, and add glucose to the sediments. So rather than adding sulfide directly, which would be really hard to do well, you just add that organic matter, and it's going to ramp up all the microbes. They're going to go crazy. They're going to reduce sulfate. So you're going to get your high sulfide. They're going to reduce sulfate to sulfide. So you're going to get your sulfide that way. So, so the experiment had 3 different treatments to it. It had a control treatment, so the seagrasses were isolated so that we could do the treatment. We had a control treatment. We had a nitrate addition, and we had a organic addition. And so the thought was that that if we added nitrate, that should reduce any kind of sulfate reduction. So we would have a control, we would have one where we'd expect nitrate reduction, we'd have less sulfide in the sediments, and then we'd have one that had all this high sulfide. So we'd have like 3 different levels of sulfide based on what we added. So again, this is the experimental design. My students, put down these cutoff buckets in the seagrass beds to isolate the areas. We added the glucose or the nitrate, or the control was done as a with nothing but just same kind of disturbance, and we looked at sulfide concentrations and then looked at biomass and productivity. So here's the sulfide data, and this is them using a little sipper to pull the pore waters out. And you can see that in both the haliduli and the thalassia beds, you add glucose, you increase the amount of sulfide. When we added the nitrate, didn't really have much of an effect. So in terms of sulfide, we didn't really see lower levels of sulfide like we initially expected. We looked at just the biomass. So at the end of the experiment, harvested the above ground and the below ground biomass. And what do we see? Well, for haliduli, didn't really seem to care. The above ground biomass looks similar. The below ground biomass looks similar. Plants weren't really worried by the fact that you had sulfide. Haliduli was fine and happy. If you look though at the thalassia, not really much change in the below ground. Definitely a significant reduction in the above ground biomass. Now it is we only had 3 replicates, so variability is an issue, but definitely a reduction there. So this is what happens in terms of biomass. So you can kind of see what it looks like inside the bucket. Seagrasses aren't really happy growing in buckets. Also, bad thing about tourists, it's a popular area. Somebody decided it would be really fun to put a bunch of hermit crabs in one of our buckets. And so there were no grasses left in that bucket. So 2 replicates, not 3. So one lesson learned, don't put your experiments in a tourist area. All right, so what about the growth rate? So again, we use that kind of leaf marking technique to try to see what the growth is. And we do see lower net production in the high sulfide treatment, maybe a bit higher in the nitrate treatment, some stimulation there. So does seem like there is some effect to sulfide. And as I said, before, we've seen high sulfides and not really seen issues. Well, last year, we had a big bloom of sargassum offshore. Maybe you guys saw it on the beaches here. Tremendous rafts of sulfide, and a bunch of it got washed in and through tidal transport ended up in Santa Rosa Sound. And this is some underwater photographs from one of our study sites, one of Lacey's study sites that we went to in June. And can you see in the left-hand photo that kind of sort of open area that's sort of blackish with the white around it? Okay, the black part is the sulfide, and the white is the Biogeochemistry, which is a sulfide oxidizing bacteria that are kind of surrounding it. So you cleared away some of the macroalgae that had gotten in these grass beds. And you could see what was happening at the surface of the sediments. And so that's the haliduli bed. In the thalassia bed, again, you can see there's a lot of epiphytes on it, and it doesn't really show up really well, but it was pretty noticeable in the thalassia beds as well. That bottom part of the thalassia picture, again, has that kind of blackish color to it, that is typical of seeing that sulfide. So under these kinds of conditions, so the macroalgae is doing 2 bad things to the seagrasses. It's covering it, it's physically on top of it, and then it's also creating this toxic environment. So I have a honors student who is working up her honors research looking at sulfide levels. Short answer is she doesn't really see changes in sulfide over a 24-hour period, but she learned a lot and got some really cool data on oxygen levels in the seagrass bed. So I have some new projects that I just started this year. I mentioned real interest in water quality in the area. Santa Rosa County has got their county restore funding. A big part of it is going towards there's a sewage treatment plant in Navarre, Florida, out at the beach. And the effluent from the sewage treatment plant, the treated effluent, goes into Santa Rosa Sound. So they have gotten money to remove that effluent from the sound. They're gonna do land disposal further north in the county. And they're also doing quite a bit of work to have septic to sewer conversion on the Gulf Breeze Peninsula. Our area, incredibly sandy soils. People have septic tanks right next to the water, basically. What do you think is happening, right? Where are all those nutrients going? So this septic to sewer conversion, is a really great thing that's going on. So I've been working with the county, starting them. We're doing a quarterly water quality monitoring program. We're doing a seagrass survey before they do all the construction, and then we'll do kind of a follow-up, after they get the wastewater treatment plant out of there. As Ruth mentioned, I'm a distinguished university professorship, which was a great honor to get, and I had to do a project. So one of the things that I really wanted to do was to kind of revisit the Merle et al. 2018 paper where he had sensors out and he did water incubations and he subtracted to get the benthic part. So what I'm doing is I'm kind of following up with that and actually doing the benthic incubations as well as the water incubations. And so we're going to do this comparison of looking at the components and how it compares to the ecosystem level measurements. And then the other part of this is the benthic flux work, which I haven't shown you any benthic fluxes. And so looking at kind of the pore water versus the benthic chamber measurements. And I always love doing nitrification work, so I'm going to do a little bit of that. And then the last project that we started in the fall is a collaboration with folks at the Florida Institute of Technology. Some of you may be familiar with issues in Indian River Lagoon. If you've heard about them having to feed manatees in Florida, Indian River Lagoon, and it's because they've lost all their seagrasses, right? So they have horrible, harmful algal blooms there. Really, awful problems with water quality. So Florida Institute of Technology is proposing that perhaps if they can reduce the residence time of water in the lagoon, it might improve water quality. Now, they're not saying this is the whole solution, but that this is a technology solution that could be helpful. And so they're developing a pilot program near Port Canaveral where they're gonna test this out in a small scale. So they have a geochemist there who's done a lot of geochemistry work, and he really started thinking that the microbes were important. And so they got in touch with us and wanted us to collaborate with them. So we've been sharing samples, comparing things, and my colleague Lisa Wadner, who does all of the microbial ecology work and all of this, we're developing the procedures to look for the different microbial transformations using qPCR. So we're going to look for the guys that are taking ammonium and turning it into nitrate, seeing if that's a significant process, particularly when there's oxygen present. Is there denitrification occurring? Is there dissimilatory nitrate reduction going on? So Lisa is doing all the molecular side of it, and I'm doing the biogeochemistry side of it. And part of what we were kind of selling in this project is that to kind of get things up and running and try to make some good comparisons. While not perfect, Santa Rosa Sound in some ways is a good analog to Indian River Lagoon. It has similar isolation from a lot of fresh water, historically abundant seagrass beds, and it's really easy for us to work there. So we're going to be in the rest of this year kind of working on this comparison of sediment biogeochemistry in Santa Rosa Sound and Indian River Lagoon. So so this idea with the flow is that if they're bringing in water from the ocean, it's going to be oxic, and so it's going to enhance this process of nitrification, and we hope to pick that up with the qPCR results. And if you don't have that going on, you have dissimilatory nitrate reduction occurring instead. So that's kind of the conceptual model for that. So lots and lots of support from this. Actually, really is not even all the students who've worked on the project, but many different folks have helped out over the years. And so I think I didn't leave much time for questions. So if you have to run, I understand. Yes.
Yeah, nice talk, thanks. I was wondering if you looked at the composition of the organic matter in the sediments as well, and looking at the differences in nitrate and phosphate in the floor water. Like, is that coming from different, that you would expect in seagrass that you would have sort of different organic matter that's being broken down to get it?
Compared to barriers, mean?
Yeah.
Well, not really, because like in Mackenzie's work, the pits were all full of seagrass blades.
Oh, yeah.
So yeah, so you definitely would see differences in quality in places with seagrasses and without. But if you're kind of near a place of the bed, it's going to be pretty similar. So you're going to get both the phytoplankton and the microalgae that sediment into the seagrass beds, as well as the more lignin, higher more refractory type material. So yeah, we do organic content just in a simple-minded way. Yeah. So the question was about organic matter and characteristics of organic matter in the sediments. Yeah. Yes, Jeff.
With your students knit genes, who's doing the nitrogen fixation, and they vary by the different. So I mean, you got maybe Raquelia, trichodesmium, diatoms, azotropes, Propospira. You know, different doing it, and are they different in different?
Yes. So we should get some indications of that. I think Lacey has optimized 3 different primer pairs, and so they are kind of different groups. And I would have to go back and kind of look up in terms of seeing what's what. So yeah, stay tuned. Yeah, she's actually working with an undergraduate, and they may have a 4th pair that they get to do as well. So yeah, I mean, and then the other thing is, you have horizontal gene transfer. So what does it really mean if it's been isolated from one? Is somebody else end up picking it up? Microbial ecologists can figure that stuff out. I'm not really going to know. Yes.
I was curious if you could tell kind of with the ray pits, like, were they recently formed, or could you tell kind of how long they've been there?
So you're asking about the formation of the ray pits?
Yeah, just if you knew like they were all fairly recent, or if some might have been around for a while.
Yes, that was definitely a limitation of the study, because we didn't actually see the rays making the pits. But she started in the the late spring, which is kind of when they're coming in. And so, kind of based on sedimentary suspension and all of that, we know that I think we know at least 1 or 2 of them were recent, because we could actually see where we saw the roots and rhizomes exposed. Right. So that one clearly had to be a recent one. Yeah. Yeah. We thought about kind of artificially making pits, and there's always trade-offs in trying to do these things. Yeah. Yes, Dottie.
So, okay, my question is, would you say that seagrasses, in our area are nitrogen limited or phosphorus limited? You know that, there's papers that say that it's phosphorus limited, but we don't have the carbonate sediment like they do in South Florida.
Right.
So. And
Right.
some of the restoration techniques with the bird steaks are saying we need these nutrients because there is evidence that they're phosphorus limited. What's your feeling?
Well, I mean,
expertise?
yeah. So, nitrogen versus phosphorus limitation in the grass beds. I mean, I think those studies that, Ken and you guys have done, do strongly suggest phosphorus limitation.
We can have it measured the sediment though yet, and actually.
But you've measured what's in the grasses. So, I think, I'm not sure there's a simple answer to it. I think that, they're very good at taking up nutrients, and I always sort of worry too about just showing the ratios, because you don't really know what the process, you don't know what the resupply and recycling is. And so that may be a little bit of a red herring, that ratio may be a reflection of how successful the plants are at taking it up as opposed to actual limitation. So I always I need to always be cautious in how to not, say something that's, you can't really know for sure. Yeah. Yes.
So you had a student who was interested in the history of the nitrogen conditions, right? Has anyone looked at the concentrations, the sulfatic concentrations over time? See how that's changed in the day since all this specialization?
No, we have no one that's really taken on sulfur. I mean, there was, one of my husband's colleagues at the EPA did a lot of, sulfur cycling kind of work. So I don't know, if you could, if you could take long sediment cores and maybe look at pyrite or something like that. I mean, the sediments are very sandy. Maybe in the channel, there's enough clays and you can maybe do pyrite. I thought about trying to do pyrite, and it's, wow, it's one more thing. And why try something else that I'm probably not going to be good at? So, yeah. So it would be interesting to do. Yeah, definitely.
That we working on. Look at it's a lot. I don't know which one it is. I can very use those. I've wanted it for a while now before I got it. I never liked it.
We'll just come by the lab on the city.
How you doing? Yeah, you're working on. Yeah, you're. A long time. Yeah.
Yeah.
I'll make some of that storm water. I was paying the overhead here. Yeah. Yeah, it is perfect. I could go kayak. It's a good fun.
Yeah. 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 Yeah, back to diving. I haven't been diving in a while.
My card. Yeah, we can. You never know there. I already had, mine is still clean from,
But it was.
Scary too, right?
It was really scary. Yeah, I had some really bad routine, but broken bones, nobody touched it. So I was the only one can ask them where those go. Yeah. Oh, it's
Yeah. It was scary. Yeah.
the aggravation behind, and I did a truck, and the truck was local too.
Where was it?
Unfortunately, range line in Hamilton, coming down here from Mobile. I was going through the green light and somebody turned left in front of me. They ran a red, turned left in front of me. I was going through the side and they smashed right into me.
Not quite that, but.
Well, that's really bad. So I just say, I got the arms. You can't drive. You're having trouble with it. Really great. Yeah, it was actually. Yeah, it was pretty. But in that run out, ran a red light, so corner.
Love running red lights.
Alive. He was like. He was driving out, I got started. Yeah.
And I can take whoever.
And where are we going? I'm sorry.
Yeah.
I don't, but I'm sure I can Google
Near
it.
the post office.
It's right underneath. Like right by the circle page. Yeah. Maybe I'll have one of you guys come with me to make sure I go to the right place.
You gotta go first. You don't want to put all of us.
Whoever wants to ride it.
Hello.
I'm Ally, PhD student.
I'm done with all my. Oh, okay. I'm not doing one right now. Yeah. Other people. Yeah.
I'm sorry. All right, yeah. We're gonna go with that. The other guy.
No, that was all. I don't know. That's easy. Right. I love the end, I love the vegetarian. Thank you. And you're okay. Well, it's not, I just talking about leaders. I mean, because she was really popular in the better center. There you go, exactly. And like, oh, that's like, you know, it was, I wouldn't just move on to that, but I would actually.
I have a 2:30 to 3:30, but I don't know if she's here or not. Yeah, I just. After that.
Yeah, I've been meeting Dr. Valentine too, and my life is very, I'm not sure what he's on. Yeah.